You know, we always hear DNA called the blueprint of life, but does that really do it justice? I mean, it's so much more than that. It's like an ancient, sprawling library, and every single book holds the secrets to building and running all of life as we know it. So, how does this library actually work? So, that's the big question, right? How can one single molecule, all coiled up inside our tiny cells, possibly manage a job that massive? How does it handle all the information needed to run you or me or a giant redwood tree? It's a challenge of almost unimaginable scale and precision. Well, it really boils down to two huge and kind of opposite jobs. First, the entire library has to be copied perfectly, letter for letter, every time a cell divides. No mistakes allowed. And second, the cell needs to be able to pull just one recipe, one single gene off the shelf to build a specific protein, right when it's needed. These two tasks, copying everything versus reading one tiny part, are constantly in tension with each other. Okay, so let's tackle that first challenge, making a perfect copy. Think about it. Every single time one of your cells divides, it has to duplicate billions of letters of DNA code, and it has to get it almost perfectly right. The molecular machinery that pulls this off, it is absolutely breathtaking. You see, this whole high-stakes copying process can't just kick off anywhere. It has to start at these very specific landing zones on the DNA called origins, and it's this super regulated four-step process. First, the right proteins get ready for the job. Then they land on that origin spot. Next, they pry open the DNA just a little bit, and finally, they call in the main copying machine to get to work. And that incredible machine is called the replisome. And it's not just one little enzyme, oh no. It's this massive, coordinated factory of sometimes more than 20 different proteins, all working together in perfect harmony to build a new strand of DNA. And inside that factory, there is one part that is an absolute superstar, the sliding clamp. It's a protein that's shaped like a donut, and it literally snaps shut around the DNA strand. This little ring then grabs onto the main copying enzyme and holds on for dear life, so it can't just float away. This simple little trick is the secret to both the incredible speed and the pinpoint accuracy of replication. And here's the really cool part. This sliding clamp thing, it's not a one-hit wonder. It is a universal solution that life figured out and has stuck with. Bacteria like E. coli use a version called beta. We humans use one called pCNA. They've got different names, they're built a little differently, but they do the exact same job in the exact same way. It's just a perfect example of evolution saying, hey, this works great, let's keep it. All right, so we've got the perfect copy down, but what about that second challenge, reading just one gene precisely? Well, a cell doesn't just read the DNA code directly. It's more complicated. First, it makes a kind of rough draft, and then that draft has to go through this whole intense editing process, kind of like a filmmaker turning hours of raw footage into the final movie. That rough draft is called pre-messenger RNA. See, when a gene is first copied from DNA, the new molecule is this messy mix of useful protein coding sections and a whole bunch of other stuff that's basically just genetic clutter that needs to get cleaned up. Now, the useful parts, the ones that actually have the recipe for the protein, are called exons. The junk parts in between are called introns. So the cell's first job is to get rid of all those introns. So how does the cell clean up this mess? It happens through an amazing process called splicing. And this, folks, is where the cell gets really, really creative with its information. RNA splicing is pretty much what it sounds like. There's this incredible molecular machine that comes in, recognizes exactly where the introns end and the exons begin, and it just cuts out the introns, and then stitches the exons together into one clean, continuous message. So the bottom line is you go from that long, messy pre-mRNA to this, a short, clean, mature messenger RNA. And this is the final blueprint that the cell can actually read to build a protein. But the story doesn't end there, not even close. Because see, the cell can get creative with its editing. It doesn't have to follow the same rules every single time. This is called alternative splicing, and it is a game-changing mechanism that lets our cells create a massive variety of proteins from a pretty limited number of genes. And just get a load of this number. This isn't some rare biological quirk. A staggering 70%, 70, of human genes are thought to use alternative splicing. This is a huge reason why we're so biologically complex. So how does it work? Well, think of a gene's exons like Lego bricks. The cell can pick and choose which bricks to use. For example, It could stitch together exons 1, 2, and 4 to make protein A. But then, using the very same gene, it could decide to use exons 1, 3, and 4 instead, creating a totally different protein B. By just mixing and matching the pieces, one gene can code for a whole family of proteins. So the cell has made its perfect custom-edited message. But making the message is only half the story. The final layer of control, the real finishing touch, is deciding exactly how long that message gets to stick around. And this brings up a really important question. Once you've made an mRNA message, how long should it last, right? Should it be a one-and-done kind of thing, used to make a protein just once? Or should it hang around for a while, churning out protein after protein? Controlling this is another super powerful way to regulate our genes. To do this, the cell adds two key features. At the front end, it gets a special protective cap, like a little helmet, and at the back end, it gets a long tail, called a poly-A tail. Together, these act like armor, protecting the message from getting destroyed right away. But once it's out in the cell's cytoplasm, it's a constant tug of war. On one side, you have these protective proteins that can bind to the message and shield it. But on the other side, you have these tiny little molecules that are actively hunting it, trying to take it down. And these tiny assassins, they're called microRNAs. They're only about 21 to 24 genetic letters long, but they are incredibly effective. They team up with a protein complex to form a seek and destroy system that zooms in on specific mRNA messages and just chops them up, instantly silencing that gene. So when you step back and look at the whole picture, you see these two incredible ways that DNA masters information. On one hand, you have replication, which is all about perfect, unchanging stability, passing that library down through the generations. And on the other, you have regulation, which is all about being dynamic and flexible, pulling just the right book at just the right time for just the right amount of time. Which leaves us with one final thought to chew on. We're always so focused on the DNA code itself, the A's, T's, C's, and G's. But maybe the true genius of life isn't just in the static blueprint. Maybe it's in the incredibly elegant, dynamic, and living systems that have evolved to read it, to edit it, to copy it, and to control it every single second.